My name is Russ Derry. I'm the Director of Education at Epilepsy Foundation of Michigan. And our topic today is understanding epilepsy at the cellular level. And we're very happy to have with us Dr. Ruggero Serafini, who is an epileptologist and researcher at Wayne State University and Detroit Medical Center. Dr. Serafini, can you just briefly describe your background and give us a quick overview of your clinical and research interests as they relate to epilepsy?
Okay. My background and my activities. I am a clinical neurologist specialized in epilepsy, but I have been, and I'm also a lab researcher studying basic mechanisms of epilepsy. Currently, I am assistant professor of neurology at Wayne State University. I have an outpatient clinic at Wayne State Detroit Medical Center. Most of my patients are people with epilepsy. I share with other neurologists at Detroit Medical Center coverage of the General Neurology Hospital Service at Harper Hospital in Detroit. I share with the other epilepsy specialists coverage of the epilepsy monitoring unit and the reading, interpretation, routine EEGs. But I spent actually most of my career and more than 15 years doing full-time basic science research related directly or indirectly to epilepsy mechanisms. I worked in several research centers. Some of them are actual centers of excellence for the study of epilepsy. The Negri Institute in Milan, the National Institute of Health in Bethesda, Washington University, St. Louis, Duke University, North Carolina. After my residency, I landed in Michigan for an epilepsy fellowship at Hereford Hospital, and in the past 3 years, I have been on the faculty at Wayne State. My research interests. I have been developing at Wayne State in the past 3 years a new assay to study basic neurophysiological mechanisms of epilepsy in brain slices in vitro, and I'm also working on a project on the interpretation of the electric waveforms of recordings of intracranial electrodes in patients that are undergoing epilepsy surgery. So that's it.
Okay, great. So the brain, as we know, is extraordinarily complex, and we certainly don't expect participants of this call to come away with a full understanding of how epilepsy in the brain works on a cellular level, and obviously even researchers only know a fraction of how the brain works. But we do, however, want this call to give people a sense of what's happening in the brain when a seizure occurs and when epilepsy develops, as compared to what happens normally in the brain. We also want people to learn more about some of the important questions that are being asked by epilepsy researchers, some of the key cellular and molecular targets that are being researched, and then how our growing understanding of epilepsy and the brain is leading to new and better treatments. So can you start by explaining what happens in brain cells, neurons specifically, during a seizure, and how that compares to the normal function of neurons?
That's a complex question that requires a long and complex answer, and I'll try to do my best to explain it in simple words that might be easy to understand even to the person who is not a physiologist, who is a professional of these things. So the basics. The brain is composed of billions of individual biological cells, the so-called neurons. Neurons are microscopic generators. Now, we are familiar, most of us are familiar with batteries. We use batteries for the car, the cell phone, the computer, some appliances. In a battery, there are two poles, a negative and a positive pole. The negative pole has an overabundance of negative charges. The positive pole has an overabundance of positive charges. You connect the two poles of the battery, and then there is a flow of electric charges between the two poles, and this can produce heat, can perform a variety of functions in the car, in the cell phone, etc. Now, a neuron is at all effects a small, microscopic, minuscule battery generator. Just the shape is different from that of the batteries we use in our daily life. The neuron is a little microscopic bag. The size is of 1/10 to 1/50 of a millimeter, depending on the type of cell. Like in a battery, there is a positive pole and a negative pole. In the neuronal cell, the negative pole, where is the excess of negative charge, is inside the membrane. The positive pole is outside. The cell, the neuronal cell, is delimited by a membrane, and this membrane is an insulating material, but at the same time, it has a molecular mechanism that redistributes the concentration of the electric charges in these body fluids. And these electric charges in the body fluids are nothing else than the salts that are dissolved in the body fluids. So there is sodium chloride, which is basically kitchen salts, potassium chloride, which is another type of kitchen salts. There is bicarbonate, like sodium bicarbonate is what you take when you have a stomach acidity, and there is calcium. So these salts are dissolved in the fluids, and when dissolved, they assume electric charges, and then when they assume these electric charges and dissolved in water, they're called ions. The membrane is not just a physical barrier that separates the internal and external environment. The mechanism establishes this asymmetry in concentrations of these particles, of these ions, is in the membrane itself. The membrane accumulates sodium, calcium, and chloride outside the cell and accumulates potassium inside the cells. Now, there are small microscopic spots in which there are tiny canals that can open temporarily for fractions of a second, and these are the channels, the ion channels, are channels that allow the flow of these electric particles, the ions, so they're called ion channels. So, trying to make things clear, I will just summarize this main point, and then I will move on to explain things further. So the main cell of the brain, the neuron, is like a battery. The negative pole is inside, the positive pole is outside. Like in a battery, you connect the positive and the negative pole, and then there is a current that flows between the two poles. The electric charges moving are the ions, the particles of salts that are dissolved in the body fluid. And in a neuron, you connect the two poles, the outside and the inside, by the opening of these canals, the ion channels, and then there is a flow of ion charges through the membrane. Now, coming to the question that you were asking, what happens during a seizure? Sodium and calcium have positive charges. There are ion channels that are permanent to sodium and open, and there is a massive flow of sodium and calcium from the outside to the inside. This causes a perturbation, an electric perturbation of the outside environment, because there are all these positive charges that flow from the outside the cell to the inside of the cell. There is a perturbation in the outside environment, and then if you put a sensor to electrical activity, to electrical field, to electrical potential outside the membrane, this sensor, this electrode, will pick up this perturbation and it will detect a waveform that corresponds, that parallels the opening of the sodium and calcium channels. So let me summarize again this point. When a sodium or a calcium channel opens, positive charges flow inside the cell. There is a perturbation in the outside, in the extracellular environment, and this can be detected by an electrode. Now, what happens in a seizure? What happens in a seizure is this. Normally, the number of cells that are synchronized, that work simultaneously in their opening of the sodium-calcium channel is relatively limited. In an epileptic tissue, there is an increased connectivity between neuronal cells. And because of this, then the cells, they open their sodium and calcium channels simultaneously. A very, very large number of cells, they open their sodium and calcium simultaneously. As a consequence of this, the perturbation in the external environment becomes extremely big, and an electrode will pick up a very, very large electric signal. If something like this happens on a surface of the brain, of the cerebral hemisphere, that is very large and it is very close to the scalp, then even an electrode that is placed outside the scalp, on the scalp, will be able to detect this large amplitude electric signal. And summarizing this main point here, activation in sodium-calcium channel can produce an extracellular electric signal. When this involves simultaneously a very large number of neurons over a large surface, the electric signal generated can be very large and can be detected by electrodes on the scalp. And now, because of this, how would one treat and how would one prevent epileptic discharges from occurring? Well, the traditional way has been that of using drugs that block the sodium and calcium channels. So they plug them, they prevent them from opening, and that prevents this perturbation of the electrical perturbation of neural tissue to happen. Now, about understanding the disease epilepsy and finding new ways to treat epilepsy basically means understanding the mechanism that lead to these increased interconnections between neurons, increased interconnections that allow this excess of synchronization. Now, epileptic discharges that are due to this synchronized opening of sodium-calcium channels in multiple cells can occur in different ways. If they occur as in a rhythmic, repetitive way, spreading from one region of the cerebral hemisphere to another region, that's when you have an actual seizure. So seizures are rhythmic, repetitive epileptic discharges. A typical seizure in the temporal lobe, for example, manifests with rhythmic waves that occur at about 5 to 8 per second, 5 to 8 hertz. Now, a seizure is also called an ictal event by epileptologists. But epileptic discharges can occur also sporadically, randomly, in an isolated way, and like the seizures are called ictal discharges or ictal event, these isolated, random, sporadic discharges are called interictal discharges. Interictal discharges are not seizures, but they still indicate the presence of epileptic excess synchrony between cells. And even if they are not seizures, they can still most likely disrupt the capacity of the brain to process information. So that's my explanation.
Okay. I am following you so far, so that's great. Does anyone else have any questions about what we've spoken about so far? Or does everyone kind of understand how a seizure occurs on a molecular, cellular level? Okay. So everyone has a seizure threshold or a level above which they'll have a seizure. And for people with epilepsy, that threshold may be chronically lowered, either by brain injury or genetic factors or other factors, or it also can be temporarily lowered by things that we think of as triggers like stress or sleep deprivation or missed medication. Can you talk a little bit about excitation and inhibition and then how these relate to seizure threshold and then what different factors affect seizure threshold?
Okay. We need to make a distinction between acute seizures and actual epilepsy. Epilepsy is a chronic condition, so you have epilepsy when you have at least 2 unprovoked seizures. Acute seizures can occur in the normal non-epileptic brain, and someone takes a shot of cocaine or is treated with a convulsant medication, then you can have a seizure without having epilepsy necessarily. A non-epileptic brain can seize after a convulsant stimulation, and we'll talk subsequently about neurotransmitters. But neurotransmitters are these chemical substances that are present in vesicles at the termination of an axon, and there are several types of neurotransmitters. There are neurotransmitters that increase the firing rate of cells, and these are called excitatory, and one of them, the typical one, is glutamate. And glutamate typically opens sodium and calcium channels, and there are neurotransmitters that have an inhibitory effect on this, and one of the common ones is an amino acid that is called gamma-aminobutyric acid. And there is an ion channel, the potassium channels, that tends to block, to reduce the firing rate of cells. And there is this balance between excitatory neurotransmitters like glutamine and inhibitory mechanisms like the gamma-aminobutyric acid and potassium channels that allow.
Just to interrupt that's commonly referred to as GABA.
GABA, yes.
Yeah, okay. Just for people who may not have heard of that term. So go ahead.
Yes. So that this allowed the normal function of the brain to process information. And you can provoke acutely seizure in a normal brain tissue by blocking GABA activity or by blocking potassium channels. And then you can have seizures. And a lot of studies in epilepsy have been done on acute seizures induced in normal animals or in normal brain tissue in vitro. But epilepsy is different. As I said, epilepsy when you have at least 2 unprovoked seizures. Epilepsy is a chronic condition. Perhaps in a chronic, and we are in the cutting edge of research in this, so I can only say perhaps there is evidence suggesting this, but there is no conclusive and definitive proof of this. Perhaps in a chronic epileptic focus, there is a center of a small size where you have this epileptic hypersynchrony, so cells that are interconnected in a high way between one another, a large number of cells that are interconnected, and this produces the epileptic hypersynchrony. But around this center of epileptic hypersynchrony, now this center of epileptic hypersynchrony is kept in check by surrounding brain tissue. Now, there is a neurophysiological phenomenon that was discovered in the 70s by Dr. David Prince at Stanford University. So now it's 4 decades ago, and it's still attracting a lot of interest. And there is still a lot of work that has been done on this, and this is called the surround inhibition. Now, what Dr. Prince found was that in an experimental epileptic focus, he found that there was a surrounding zone that was resistant to epileptic activity, and then he found that in this surround inhibition zone, the surrounding, there was an enhanced GABAergic, gamma-aminobutyric acid activity. So perhaps it is the collapse of this surround inhibition that allows the spreading of epileptiform activity. So in this way, perhaps there might be a transition from isolated sporadic discharges, the so-called intellectual discharges, into actual seizures. But this is just an educated guess. In the end, I have to admit I'm not able to give a strong answer. I may possibly try to give an answer to some possible trigger facilitators of seizures. For example, catamenial epilepsy. There are ladies in which seizures typically occur at the time of their menstrual period. Now, before the menstrual periods, there is a drop in the serum concentration of progesterone. Now, progesterone has been known now for decades. Progesterone and its metabolites are known to interact with the receptors for gamma-butyric acid, for GABA. So if there is a change, if there is a pronounced drop in the progesterone concentrations in the intercellular space, this drop of a drug, of a substance that stimulates the GABA receptors that have an inhibitory effect, this perhaps could be the mechanism that is behind the increased susceptibility to seizures at the time of the menstrual period. How sleep deprivation facilitates seizures, I have to admit I don't even have, not even a possible mechanism in my mind. I found it interesting that seizures have a circadian rhythm, and by just making a jump of imagination on this, just a flight of imagination, I thought that perhaps there might be a circadian rhythm hormones that could be like progesterone during the menstrual period that drop at the time of the time in which basically if you just have sleep deprivation, or the time in which you go to sleep, and that would cause you to have seizures when you fully sleep. And I have to admit, however, that I do not recall any study on this. There may be, but I just am not familiar with that.
Okay. And is there any clues as to why stress may be a trigger? Is that related to cortisol or any kind of impact of cortisol on neurotransmitters? Or
Glucocorticoids are pro-convulsant. There are receptors for glucocorticoids. Actually, there are several types of receptors for glucocorticoids in the brain. So there are the typical steroid hormone receptors that are in the nucleus, and so they have the steroid has got to get inside the cell, has to bind the receptors in the nucleus, then the nucleus has to change the transcription, enhance or depress the transcription of some genes. This, however, takes several hours to be effective. There are, that I have read, also receptors on the membrane. I am not familiar, however, about what type of receptors, what type of mechanism they would actually have. The bottom line, however, is that glucocorticoids are pro-convulsant. So if you would have an increase in glucocorticoid, that might end up basically resulting in increased susceptibility to seizures. But I don't think this has been actually studied in a detailed way in any way.
Okay. So let's talk about some of the different types of anti-seizure medications and how they work on a cellular molecular level. And for this, we'll need, again, a little more introduction on the concepts of neurotransmitters and ion channels and other molecular targets.
I talked before about ion channels, and I've already mentioned briefly about neurotransmitters. Let me just present some additional details. So a neuron is made of a central part that is called the soma or the cell body. And from the soma or cell body, there are small processes that branch out from this cell body, like the branches of a tree. These are dendrites, small processes that come out of the cell body. Then there is a long process that represents a sort of cable. This is the axon. The axon conducts electrical activity from one neuron to another neuron. There are different pathways. There are, for example, pathways on the surface of the cerebral hemispheres, on the cortex, and there are other pathways that start from a bunch of cells that are deep in the brain, so the so-called basal ganglia, the thalamus, and then they send their axons on the cortical surface. I'll talk about this later on in more detail. And there are also processes that there are cells that are on the surface of cerebral hemispheres that project to the thalamus and the basal ganglia. And at the end of axon, there are small vesicles that contain chemical substances, the neurotransmitters. I already mentioned that there are amino acids like glutamate, aspartate, gamma-butyric acid, that is GABA, monoamides, dopamine, adrenaline, or adrenaline, serotonin, peptides, small proteins. When the electrical activity gets to the end of the axon, it determines the fusion of the vesicles with the membrane of the axon, and the neurotransmitter that is present in these vesicles is thrown outside the membrane, and when it is thrown outside, it can bind a molecular counterpart, the receptor, that is located on the surface of an adjacent neuron. Each of the different chemicals have its own receptors, and actually each chemical, each neurotransmitter can have many types of receptors that can have selective sensitivity to various drugs. That is a topic that pharmacologists are interested the most. And when the neurotransmitter binds to the receptor, this can trigger a change on the membrane of the adjacent neuron, and that can trigger the opening of other ion channels. And the opening of ion channels, that is the terminal neurotransmitter at a synapse, at this junction between an axon and another cell, is called a synaptic potential. Synaptic potentials are somewhat slow under neurophysiological standpoint, slow. So they last several milliseconds, dozens of milliseconds, or hundreds of milliseconds. The ion channels that they open on an axon when the electrical activity propagates along the cable are very fast, milliseconds or fractions of milliseconds. And in the last 30 years, there has been an enormous impulse towards the characterization of these processes because they discovered, two German scientists, Sackmann and Herr, they discovered a measurement technique that allows the direct recording of the opening and closing of single ion channels. And having the possibility of studying the activity of a single ion channel has been an enormous revolution in biosciences. Now, let's talk about anti-seizure medications. Many of the drugs that are used to treat epilepsy are sodium channel blockers. So Dilantin Tegretol, trileptal, lamital, their main effect is thought to be due to a block of sodium channels. And even other drugs, such as, for example, topiramate, Topamax, or Zonegram, zonisamide, a component of their mechanism of action consists in blocking the block of sodium channels. Now, an important point is this, that these drugs are more effective in blocking sodium channels when they open in a repetitive way. So if there is just one or two openings that are isolated, then the effect of these drugs will be minimal. But if there is a repetitive, ta-ta-ta-ta-ta-ta-ta, like when during a seizure, then the effect of this drug is the most pronounced when the sodium channels open in a rhythmic repetitive way, like, for example, during a seizure. So this is interesting because these drugs have a somewhat limited effect on the normal cerebral activity. It's not that they don't have any effect on the normal cerebral activity. They still have, but it is limited. Their maximal effect is when the sodium channels open in a repetitive way during a seizure. And so that's allow them to have a selective effect on seizures and a relatively minimal effect on normal cerebral function. Other drugs, Keppra, levetiracetam, seems to affect a protein that is involved in the release of neurotransmitter from the synaptic vesicles. When the electrical activity gets to the distant extremity of the axon, so then the vesicles that contains the neurotransmitter, they fuse with the outer membrane of the axon, and so then the contents of the vesicles is thrown out. And so Keppra somewhat seems to bind to one of these proteins that is involved in the fusion of vesicles with the membrane. Lacosamide is a relatively recent drug, Vimpat basically. It's a blocker of a sodium channel, but seems to be a different sodium channel than that is blocked by the lantin, Tegretol, and trileptal. Now, so far, what I've mentioned was this. Drugs that block sodium and or calcium channels. But these drugs, however, none of them affect the initial mechanism of the disease, that is, the increased interconnection between neurons. So these drugs do not affect the mechanism that established the increased interconnection. So somewhat they're able to control the symptoms of the disease, but they do not treat the disease in itself.
Right, and that's one reason why
yes.
They used to be called anti-epileptic medications, and now we're moving towards calling them anti-seizure medications because they don't really treat the epilepsy. So the type of seizure a person has depends on the location of the abnormal electrical activity in the brain and then how widespread it is. Can you talk a little bit about how seizure activity spreads? For instance, how like a simple partial seizure spreads to become a complex partial seizure, or how it might become a generalized tonic-clonic seizure, or maybe how, and then compare that to how a primary generalized seizure would just rapidly swamp the whole brain without any clear starting point. So can you talk a little more about the spread of epileptic or seizure activity?
Okay. Now, the first two questions. How a simple partial seizure becomes a complex partial seizure. How a simple partial seizure becomes a generalized seizure. There is in some way a common denominator, a common feature in the answer to these two questions. So I mentioned a few minutes ago about the fact that there are different pathways for the cable, the axon, that starts from a cell. So there are cells, there are neurons that are located at a deep part of the brain, in the thalamus or in the basal ganglia. There are other neurons that are located in the brainstem. The brainstem is that part of the brain that connects the cerebral hemispheres with the spinal cord. It's a sort of upper prolongation of the spinal cord. Now, in the brainstem, so on the very, very top of the spinal cord, and a little above that, in the cerebral hemispheres, but deep in the cerebral hemispheres, in the thalamus, there are cells that send their axons towards the cortex, towards the surface of the cerebral hemispheres. And these cells in the brainstem and in the thalamus they have a spontaneous electrical activity. They are somewhat like those parts in the heart that make the pacemaker. So they beat spontaneously. By beating spontaneously, then they propagate the activity to the other muscle cells. So the brainstem and the thalamus are the pacemaker of the brain. They have a rhythmic, spontaneous electrical activity, and then they propagate by their action to the surface of the cortical hemispheres. Now, probably they thought that this possibly might be the neurophysiological correlate of the alertness. So they maintain in activity the cerebral hemisphere by propagating continuously electrical activity to the cortex. And now, during sleep, what happens is that this pacemaker activity of activation of the cortex that comes from the thalamus, the brainstem, is shut off. And complex partial seizures, there have been in the past 10 years a number of publications on this. They are complex partial seizures because in some way, like similarly to what happens during sleep, they turn off this activating function of the thalamus and the brainstem. And so basically they reproduce a situation similar to that of sleep. So that's how a simple partial seizure might end up becoming a complex partial seizure. Now, how does a simple partial seizure becomes a generalized, like for example, a generalized tonic-clonic seizure? There have been decades of data on animal models. Now, on this, the transition between a seizure that is limited to a small surface, more cortical surface, into a generalized surface, to a seizure that involves the whole surface, might not necessarily go through an expansion through the surface of the hemispheres. Probably, most likely, there is an involvement also of the subcortical nuclei. So the focal epileptiform activity in a small region of the brain in some way activates the thalamus, and in the thalamus then sends an increased spiking and increased electrical activity throughout the whole cerebral hemisphere, through the surface of the whole cerebral hemispheres. So that would be like the common feature between the two questions that you ask: how a simple partial seizure become a complex partial seizure, and how a simple partial seizure becomes a generalized seizure. There may be for both questions similarly an involvement of subcortical nuclei. Now, about the question of where does the seizure start in idiopathic generalized epilepsy? Here, perhaps the distinction between focal localization-related epilepsy on one side and idiopathic generalized epilepsy on the other is not entirely clear-cut. Perhaps the initial trigger for generalized idiopathic epilepsy might be a focal cortical lesion. There are epilepsy cases that are definitely focal localization-related epilepsy that exhibit diffuse spike and wave discharges, and this is a case in which perhaps there might be again an involvement of the thalamus. There have been in the past years some molecular biology genetic studies that shows that at least some cases of juvenile myoclonic epilepsy are associated to an abnormality of the molecular components of the GABA A receptors. So perhaps the cortex has a loss of inhibition, and so subcortical impulse from the thalamus might end up becoming epileptogenic. However, this is just speculation. Some of the drugs that are used to treat idiopathic generalized epilepsy, such as Depakote or ethoxylimide, they have this feature, this molecular mechanism. They seem to have an effect on the pacemaker function in the thalamus. So in some way they suppress, they decrease the pacemaker function of the thalamus. However, that does not mean necessarily that the initial trigger, the culprit, could be in the thalamic pacemaker. So there might be like a small focal lesion, perhaps in the mesial interhemispheric cortex. Then there will be an involvement of the thalamic pacemaker, and the thalamic pacemaker then sends projections to the whole cerebral hemispheres in the cortex, both cerebral hemispheres, and that would cause the generalized spike and wave discharges. So you can have a therapeutic intervention, perhaps by decreasing this pacemaker activity in the thalamus.
Okay. let's talk about some of the different other treatments besides medications for epilepsy, and how those might work from a cellular perspective. So, looking at epilepsy surgery, for example, how in many cases it may be removing scar tissue, but it also might be creating new scar tissue where the incision is made. So how does that actually end up working from on a cellular level?
In epileptic focus, so in a region of the brain in which there is this hypersynchronous activity, is not a mathematical point that doesn't have any sides. It must have a spatial extension. So probably, most likely, it represents a network of cortical regions that are coupled to one another. So separating the epileptic focus also from the non-epileptic tissue may not be easy, because there is a continuity of one into the other. Now, in an epileptic focus, there probably is a pacemaker. So I mentioned before that there is a pacemaker in the thalamus and the brainstem, but most likely in an epileptic focus there is also a pacemaker in that region. So there is a region in which the process of excess synchronization is initially started, like the primum movens, the initial trigger. That's where there is a pacemaker. Then this propagates to the adjacent cells, and so then you have a propagation zone. And then there is the symptomatogenic zone, the zone of the symptoms. The fact is this: the whole brain that we have, when people do a resection, a number of times they may not have any neurological deficit at all. This is puzzling, because it looks like that you could remove a large part of the brain and you will still be able to function very well. Yet there are specific zones of the brain that have a well-defined function. So if you remove it, then you'll not be able to talk. You will lose sensation on one side of your arm. You'll be paralyzed in your arm, and so on. So this is the so-called eloquent cortex. And I'm mentioning this to explain the meaning of the word the symptomatogenic zone. A seizure may occur in a part of the brain that does not have any immediate, obvious, direct function. It might start in that region, it might propagate, but then it becomes symptomatic when finally the propagation of epileptic formativity gets to the active eloquent cortex. And so then that's the symptomatogenic zone, is the part of the epileptic tissue that somewhat is involved causing symptoms. Now, a resection, resective surgery, may disrupt the pathway that leads to the involvement of the symptomatogenic zone for simple partial seizures. That is a possible mechanism. Or alternatively, resective surgery may interrupt the pathway that leads to the generalization. So the resection in some way may end up touching some of these axons that goes from the cortex to the thalamus, because the generalization consists most likely in a mechanism in which the thalamus sends throughout the whole cortex on both hemispheres just this epileptic form activity. So somewhat if you interrupt the activation, the pathway that leads to the thalamus activating the rest of the cortex, then basically you prevent the generalization. Now this comes to my mind because there are numerous cases in which a resective surgery allows a complete resolution of the symptoms. But there are also many cases in which the outcome is favorable, but the patient will continue to have simple partial seizures. He may not have generalized seizures anymore, but will still have sensations like a creeping, crawling sensation on the arm, like a somatosensory seizure, or we have experiential auras with déjà vu and déjà vu. So in such cases, if the patient continues to have auras, it is the pathway of generalization that is actually being disrupted by the resection, rather the actual cortical surface. To my knowledge, there has not been much attention paid to this. scar, gliotic tissue, and epileptogenesis, because you were asking, the surgery may remove the scars, but it also may be creating new scar tissue. That is a very interesting question, and I don't think this has actually been studied, at least not been studied extensively, at least that I know. Okay. I have often suspected that scar tissue by itself may not necessarily be epileptogenic. It is the traumatic process of doing a resection that then triggers a mechanism of epileptogenesis in the surrounding tissue, and then the scar tissue, the gliosis, may be a side product, may not be necessarily be epileptogenic itself. In the 80s and in the 90s, there are still people that are working on this, but there has been a lot of skepticism on this hypothesis. Glial tissue is able to synthesize a metabolite of tryptophan. Tryptophan is amino acid, is a metabolite of tryptophan. It is called quinolinic acid, and quinolinic acid is a drug that binds to the glutamate receptors. And so people have thought, well, that could be possibly the mechanism by which gliotic tissue, it releases a substance that opens glutamate channels, and that perhaps it might facilitate seizures. However, the main objection to this theory was that it takes very, very high concentrations of quinolinic acid to provoke seizures. So.
And just while you mentioned that, it leads to one of the other topics I was going to ask about, which is most of when we talk about brain cells, we're usually focusing on neurons, but there are other types of brain cells as well, astrocytes and other glial cells, or I don't know if it's glial or glial, but and they may play a larger role in seizures than once thought. You mentioned one example of how they might be involved. Are there other ways that are being studied how these other brain cells besides neurons can be involved in epilepsy?
Astrocytes are a type of glial cells. Astrocytes make the structural skeleton of the central nervous system. However, besides being a structural mechanical support, they also have other roles. So astrocytes can regulate the chemical composition of the extracellular space. So they can regulate potassium concentration, and they can uptake glutamate and aspartate. Now, there are papers that have been published that shows that in mesial temporal lobe epilepsy, the astrocytes are not able to regulate extracellular potassium concentration, and this seems to be specific of epileptic foci of the mesial temporal lobe. And so if you take astrocytes from someone that, from a human being that, for example, for tissue that's been removed because of a cancer or other things, then the capacity of regulating potassium concentration is normal. But if you take the astrocytes of an epileptic focus, then these astrocytes cannot regulate potassium concentration. That's an interesting finding, because then if you have an increase in the extracellular potassium concentration, there is a cascade of other processes then that can disrupt also the regulation of other ions, and they can make cells more excitable, and so on. And I mentioned before about quinolinic acid, and there is a pathway, biochemical pathway, that refers to the metabolism of tryptophan, and these metabolites of tryptophans are called kynurenins, and there are excitatory and inhibitory kynurenins. Quinolinic acid is an excitatory one. It opens glutamate channels, and there are other ones, like, for example, quinurenic acid, that is a blocker of the glutamate open channels. These are not, however, neurotransmitters. These are chemical mediators, and they are produced by astrocytes. An interesting line of investigation. This, however, has been presented at meetings, but has not been published yet. Dr. Jeff Slope's laboratory, so he has known in here at the Wayne State. So he has this line of investigation in which he makes histochemical molecular biology analysis studies of tissue that is resected in individuals that have drug-resistant epilepsy and undergoing epilepsy surgery. So what he has found is that in areas of the brain that have a high frequency of epileptic discharges, there is a type of glial cells, they're called the microglial cells. And so microglial cells are inflammatory immunitary cells. What do they do over there is not really very clear. There is a bunch of studies that have been done in the past 10 years about inflammation and epilepsy, and perhaps they might secrete like mediators of inflammation. But so this is an exciting, interesting topic, but it's just the very, very beginning.
Right. Okay. Another treatment option, just was curious about what we know about how it affects cells, is vagus nerve stimulation, and then also other neurostimulation-based therapies. How do those affect the function of neurons?
Well, I can present an hypothesis based on what just mentioned before. So we talked about brainstem and the thalamus. There are neuronal cells, and their axons goes to the surface of the cerebral hemisphere, to the cortex, and so they are responsible of the activation of the cortex, and perhaps most likely they also participate in the process of generalization of simple partial seizures into generalized seizures. And so the vagus nerve originates from the lower brainstem. So the brainstem, as I said, is connected to the cortex by these neuronal pathways. So we know that the brainstem can modulate cortical electrical activity. So conclusion, perhaps the vagus nerve stimulator, this affects these connections between the brainstem and the cortex, connections that allow the brainstem to activate the cortex. I know of one study, there is not really much published on this. I know that there was a study that was done in Italy in 2004, and it used this technique, transcranial magnetic stimulation, and you apply a magnetic field on the surface of the scalp, and this somewhat affects the function of the neurons on the surface of the cerebral hemispheres on the cortex. And then you can use different type of protocols of stimulation, and some of them can actually assess also for the presence of intracortical inhibition, which supposedly to be GABA or potassium channels. So the study that they published is a series of 5 patients. 4 of them, and all of them had a vagus nerve stimulator implanted, and 4 of these patients had an improvement in the seizures. And all of these 4 patients with the improvement of the seizures after the placement of the VNS, they had an enhanced inhibition in the cortex. One patient had very, very little or minimal improvement, and this one had no enhancement of the intracortical inhibition. To my knowledge, this is the only study that somewhat tries to get some insight on what are the mechanisms. Other neurostimulation-based therapy, there is the neuropace. Has been extensively investigated here in Michigan, actually, at Danforth Hospital. So, in some individuals that have drug-resistant epilepsy, and in which it has been that these individuals are not candidates for having a resective surgery, but in which it was possible to define where is the epileptic focus, where the epileptic form activity has been originated. Already 20, 30 years ago, they started to attempt this modality of treatment, placing an electrode in the cortical region that is the source of the epileptic form activity, and this electrode will provide recurrent electric impulses, and probably what happens is that these external electric impulses that are provided somewhat determine a desynchronization of the cells that otherwise they would fire together. And there about 1 or 2 years ago, actually more than that, I think it was 3 years ago, the Food and Drug Administration approved the deep brain stimulator for the treatment of epilepsy. So with the deep brain stimulator, you put electrodes. This is a technique that is used only in drug-resistant epilepsy when you cannot perform a resective surgery. So some individuals might have a benefit by the placement of electrodes in the thalamus, and so in some way perhaps stimulating the thalamus, then you disrupt the pathway through which the thalamus determines a generalization of a seizure.
Okay, great. And then lastly, as far as treatments, the ketogenic diet, how does that affect neuronal function?
To the best of my knowledge, the anticonvulsant effect of the ketogenic diet works in children. To my knowledge, there is no evidence this works in adults. To the best of my knowledge, the anticonvulsant effect coincides with the appearance of high blood levels of ketones. There is a study of 2003 that shows that acetone, which is a type of ketone, has an anticonvulsant effect on animal models of epilepsy. And to the best of my knowledge, the papers that I have reviewed, that I have read, that presentations that I have been at conferences, to the best of my knowledge, ketones do not have a direct effect on the receptors of glutamate of GABA, so they do not affect directly excitatory or inhibitory neurotransmission. Do not affect directly, so they don't interact with the receptor. But there are data that suggest that the metabolism of glutamate is affected by ketones. So, glutamate is an excitatory proconvulsant neurotransmitter. So, somewhat, if you reduce the amount of glutamate in the intercellular space at the synaptic levels, then you can have an anticonvulsant effect. Another hypothesis is this. Now, ketones are the metabolic precursor of cholesterol. So the metabolic pathway that leads to the synthesis of cholesterol starts with ketones. And cholesterol is the precursor of steroids. And I mentioned before that progesterone interacts with the receptors for GABA. Progesterone and its metabolites. So there are several steroids that are modulators and inhibitors of the GABA receptors, and so perhaps a ketogenic diet might enhance the synthesis of cholesterol, leading to an enhanced synthesis of steroids, and so that would be a possible mechanism of action.
Okay. In terms of new treatments that are being studied, looking at new molecular targets, are there many new molecular targets that are being discovered and actually translated into treatments, or are most treatments kind of just variations of existing treatments?
Well, I think the past 10 years have been exciting for the study of new mechanisms and new therapies for epilepsy. About 8 months ago, a new drug was approved by the Food and Drug Administration. It's a drug with an entirely different mechanism of action. Glutamate receptors. There are several types of glutamate receptors, and they are named by compounds that specifically bind to them. One of them is called the AMPA. So there is a subtype of glutamate receptor. It is called the AMPA receptor. And this drug that was approved by the Food and Drug Administration is called perampanel, and it is the first blockers of the AMPA receptors that we have available. So this is new. Then potassium channels, about a year and a half ago, also approved and are actually used in some patients. There is the first drug, to my knowledge, that is an actual opener of potassium channels, is called Potiga. The previous name, when there were clinical studies that were ongoing, was retigabine. So some that might have followed the literature might know that by the name of retigabine. So GABA is the inhibitory anticonvulsant neurotransmitters. GABA has a multitude of different type of receptors. There is a work that has been ongoing for the past decades, mostly in Switzerland, however, defining the different subtypes of GABA receptors and identifying compounds that bind to some of these subtypes of the GABA receptor. So this might be interesting, because then you could have an anticonvulsant drug that has a very little limited sedative effect, or you could have like the muscle relaxant drug that doesn't have a sedative effect. So that is promising. I mentioned before that progesterone is a hormone that also interacts with the GABA receptors. So there are new drugs that are in the pipeline that are steroids and that interact with the binding site of progesterone on the GABA receptor. And one of them is called ganaxolone, and I think it is close to approval by the Food and Drug Administration. There have been studies on diuretics as anticonvulsants. Now, as I mentioned before, the membrane separates an internal from an external environment in a cell and establish an asymmetry, a difference in concentrations of different ions. Well, that's basically what diuretics do. That's how they work at the level of the kidney. So they move ions from one side of the membrane, the kidney, to the other side of the membrane, so that they are eliminated and then they go end up in the bladder together with water. But they have a similar effect also at the level of nerve cells. So they can redistribute chloride concentration. Now, in animal models of epilepsy, there is one of such drug. It is called bumetanide. It's a drug that is being used in humans as a strong anticonvulsant effect. About 2 years ago, there was a clinical trial in Massachusetts General Hospital about the use of bumetanide in status epilepticus in neonates. I think the study then was shut down because they were not recruiting enough patients, and not because they were not finding it was not working, just because they could not recruit enough patients. So I talk also about ganaxolone. One thing that I would like to point out is this. Now, I mentioned several new drugs. Some of them are actually already available. Others are going to be available. Now, individually, each of these drugs, taken by itself, taken alone, may not necessarily have a better effect than the old drugs. However, there is an enormous advantage in having drugs with a different mechanism of action, because combining drugs with a different mechanism of action, you can achieve a synergism. So if you take a drug with mechanism and another drug with a mechanism, you can have the two different mechanisms working together, potentiating each other. So we have the so-called rational polypharmacy. Polypharmacy, multiple drugs that are used in a rational way. And using a rational way means combining drugs with different mechanisms of action. Don't give to people that have drug-resistant epilepsy combine drugs that have the same mechanism of action, but rather combine drugs that have a different mechanism of action. Rational polypharmacy has been studied extensively in animal models, but there are data that are showing that also in human epilepsy, some of these rational polypharmacy combinations have a very, very powerful effect.
Very good. Okay, so we've talked about what happens in the brain during seizures and then treatments designed to prevent seizures. I wanted to move on and talk a little bit about epilepsy and then what some of the different cellular, structural, and genetic causes of epilepsy are. And we'll also talk about epileptogenesis, which is the process whereby the brain develops epilepsy. And then what, if anything, can be done to halt or slow this process. Before we do that, did anyone have any quick questions about anything that we've discussed so far about how the cellular mechanisms of seizures? Okay. So let's start by talking about some of the different causes of epilepsy and how they translate on the cellular level to a tendency to have seizures. So first, can you talk a little bit about some of the genetic causes of epilepsy, like what are referred to as channelopathies, and how they lead to epilepsy?
Okay. Well, let me say, genomics of epilepsy is a field in which there is a lot of research that is ongoing. There has been some findings, definitely, that have been very important. However, it's very difficult to conclude, let's say, if we find that there is in a family, there is a mutation of this or of that ion channels. It's not easy to correlate that to a mechanism of excitability, because if you have an abnormality of a gene, that abnormality, the genetic abnormality, can also lead to changes in the development of the central nervous system. So then we need to distinguish the effect on neural development from the effect of the genetic abnormality on epileptic hyperexcitability. In some conditions, it has been possible to figure it out that the ion channels that was mutated, was abnormal, leads to epileptic hypersynchrony. And the most well-known cause is the most well-known disease is a disease that is called severe myoclonic epilepsy of infancy, that was characterized by a French epileptologist, Charlotte Dravet, and is called now Charlotte Dravet. It's called the Dravet syndrome. Children develop seizures with fever, and the seizures are very difficult to treat with drugs, and there are also abnormality in coordination. Many cases of Dravet syndrome have been shown to be related to a genetic abnormality of sodium channels, and in these conditions, people should not be treated with the classical anti-epileptic drugs that are sodium channel blockers. So Tegretol, Trileptal, Lamictal cannot be used because in these individuals, the sodium channels are already disrupted, not working well. An interesting point is this, that an animal model of this disease has been made. So with molecular biology technique, they obtained mice that have the same genetic abnormality of Dravet syndrome with the mutation of the sodium channel. And these animals, when they are placed in a warm bath, so trying to mimic the condition of a child developing fever, then they develop seizures like the human subjects. And in cellular studies, when they record the opening and the closing of ion channels, and they have figured it out that they are extremely sensitive to changes in temperature. So when they record at 32, 33, 35 degrees centigrade, then the function may be normal, but when the temperature of the recording system gets to 39 or 40, then the function of these sodium channels is totally disrupted. And I know about genetic abnormalities that have been identified in families with idiopathic generalized epilepsy, and I think I mentioned them before, that there is a decrease in the intracortical inhibition. The role, in my view, in this case is less clear, because we should have an animal model to actually test whether this mutation has direct effect on cell excitability.
Okay. Now, without going into too much detail, can we talk a little bit about neuronal migration disorders? This is where basically during development the neurons don't go where they're supposed to, and the resulting structural abnormalities, and then how they can lead to epilepsy.
There are several types of neuronal migration disorders. There is the periventricular heterotopia, the subcortical laminar heterotopia, the polymicrogyria, and the lissencephaly. Some of them have a genetic basis, others don't have that, and probably are due to local strokes, local infections, an environmental factor. The fetal central nervous system has a very simple structure. It is a tube, basically. And this tube has a wall, and then inside the tube there is a fluid. So that's the primitive central nervous system. During the maturation of the brain, the cells that make the wall of the tube proliferate, and when they proliferate, then they migrate towards the outer surface of the wall of the tube. And at the end of the embryonic development, the cells that are located in the outer part of the wall, they make the surface of the cerebral hemispheres, the cortex. The internal space of the tube makes the ventricle, and that is a space that is filled with the cerebrospinal fluid. Now, what are the structural abnormalities? In some cases, some of the cells do not migrate entirely towards the outer part of the cell, so they lag behind. They remain stuck near the internal cavitary space. So when the brain is finally mature, they remain near the ventricle. So that is the periventricular heterotopia. Hetero means abnormality. So heterotopia, and it is a local abnormality, and it is around the ventricle. That's the periventricular heterotopia. In other cases, the migrating cells, instead of forming the typical histological structure of the cortex at the 6 layers, they make an additional layer. So that is the subcortical laminar heterotopia. Then in the normal brain, the surface of the cerebral hemispheres, the cortex, shows ondulations that have hills and valleys. So the hills are called the gyri, and the valleys of the surface of the brain are called the sulci. If there is an excess of these ondulations, then there are many small hills, many small gyri, and this is called polymicrogyria, from Greek poly, many, micro, small, and gyri, many small gyri. And if there is no ondulation, then there is a smooth cortex without ondulation, and it is called lissencephaly. Now, how do these abnormalities lead to epilepsy? Now, my understanding and of the patients that I've taken care of, the review the literature that I have done, is this, that these anatomical abnormalities are not necessarily epileptic by themselves. Rather, they become people that have these abnormalities, they have epilepsy because these abnormalities, they are associated to focal cortical dysplasia. Focal cortical dysplasia is a histological abnormality in which the neurons have an abnormal shape. And in some of them, for example, they are balloon cells, and there is evidence that focal cortical dysplasia is epileptogenic. How, under what respect, what ion channels, what type of synaptic connections, this has not been discovered yet, however.
Mm-hmm. Okay. And one of the most common types of or causes for epilepsy are what we would call lesions, and they can be caused by stroke or AVMs or tumors or cerebral palsy or traumatic brain injury. How does a lesion affect brain cells, and how does that lead to epilepsy?
Difficult question.
Yeah.
Well, we can rely on animal models to make educated guesses. Now, in cases of a traumatic head injury, or when there is an intracranial bleeding, for example, to an arteriovenous malformation, so there is blood around neuronal cells. So blood consists of red blood cells. The red blood cells contains hemoglobin, which is a big molecule that at its center contains iron. Now, so when there is blood, there is going to be iron that is released in the intercellular spaces. Iron is epileptogenic, so there is evidence in animal models that iron is epileptogenic. And a known animal model of epilepsy is this: injection of a small, tiny volume of iron salt inside the cerebral surface of a rat. This, in the end, results in the development of chronic recurrent seizures. So injections of iron represent in some way a model of epilepsy caused by head injury, because in head injury you have extravasation of blood and also rupture of an arteriovenous malformation. Now, a strange and interesting finding is this: the animal will develop epileptic foci, but with this injection, the epileptic foci develop at a distance from the site of injection. So it's not the iron itself that causes directly the seizures and the epileptiform activity. Something so far still uncharacterized changes the neurophysiology of the surrounding tissue, something not yet characterized. In some way, however, there is a reorganization of the cortical cerebral circuitries. Now, this has come to to my attention, to my interest, because of this. One of the common vascular malformations that cause seizures is the cavernous angioma, cavernoma. And with the cavernoma, there can be small recurrent hemorrhage around the vascular malformation. And the epileptiform activity in cavernoma often originates at a distance of many centimeters away from a lesion. So it's necessary to do not just when people that have these lesions that undergo epilepsy surgery, they don't need just a simple removal of the vascular malformation, but they may need a larger resection than just the removal of the lesion. So the fact that there is epileptiform activity many centimeters away from the blood somewhat is reminiscent, somewhat similar to what happens when you inject like iron in the cortex of the animal. How can a stroke, like an ischemic stroke, cause seizure? So I talk about blood, like a hemorrhagic stroke, but how can an ischemic stroke cause seizure? We know that during ischemia, there is a massive release of glutamate from neurons. So at least acutely after an ischemic event, then there is raw glutamate. Glutamate opens sodium and calcium channels, and that can be the trigger of the cascade that leads to excess synchrony of neurons. However, with an ischemic stroke, you need to distinguish the seizures that occur at the time of the stroke and then the seizure that can happen months or years down the road. Well, perhaps at the end of this talk we might talk about epileptogenesis, because the question is, how do you go from an acute seizures in a normal brain to a tissue that becomes abnormal in that it generates spontaneously unprovoked seizures? So that is a big question.
Right. So that's a good lead-in to that question. It's obviously a complex process, but do you have what do we know so far about epileptogenesis and what type of research is being done to both better understand the process and then hopefully develop interventions to prevent or slow epileptogenesis in response to some risk factor? So if someone had a stroke, you could give some medication to stop the brain from becoming epileptic, or after if someone has a certain genetic risk factor or has a traumatic brain injury, you could do some sort of intervention to stop that process. Where are we now in terms of our understanding and the research that's being done on that?
Well, I have to thank you for this question also, because this is something that I am personally interested in. The research that I'm actually developing this time is somewhat related to this. So let me, however, talk about in general ideas that are behind this, trying to answer this question. As I said before, we know from neurosurgeon that one can remove parts of the brain surface from the cortex without an obvious impact on neurological function. And a seizure becomes symptomatic when the excess synchronization involves the eloquent cortex. Now, it's possible, it's conceivable, or even it's likely that seizures in the beginning develop in some area of the cortex that are not eloquent cortex. The disease may develop for many years, and it becomes symptomatic only when it is at an advanced stage, when the spreading of hypersynchronization it then ends up involving also the eloquent cortex. So by the time that becomes symptomatic, the process is already big. It's like an adult, but we want to understand things during the embryonic development or when it is a baby. Now, a finding that I found very interesting was an observation. It was done on brain slices, so observation on epilepsy recorded in vitro. It was done in early 2000 by a group at Georgetown University. And this has been picked up again in the past few years, and I have been reproducing this finding several times. What these researchers found was that when they stimulate the slices with the convulsion, the brain slices, the epileptiform activity does not involve uniformly the cortical surface of the slice. Rather, there are small microscopic discharges of the size that are inferior to that of a square millimeters. So these are microscopic discharges. So if this has been, the presence of micro discharges has been demonstrated a few years ago, so in human brain. And when they happen in an isolated fashion, most likely these are asymptomatic, and they are very small, and they may possibly represent the very beginning, the initial seed that in the long term ultimately leads to the establishment of a large epileptic focus that is symptomatic. It is interesting also that this can be studied in vitro. Now, the cortex, the surface of cerebral hemispheres, is organized into the repetition of modules, of modular units. These modular units are the so-called cortical columns. A cortical column is a part of the cortex that has the size of approximately 0.3 millimeters, and it has all the cells that are making the cortex. So the cortex is actually made of the repetition of this unit. An animal with a big brain has more cortical columns. An animal with a smaller brain has fewer cortical columns. So, but the size and the structure of a cortical column is the same. So the hypothesis that has been advanced several times is that at the very beginning of epileptogenesis, there can be a cortical column that becomes hyperexcitable. So if you cut down the problem of what happens, and then you cut down to a very, very simple unit, then the problem becomes easier to understand, rather than dealing with like a big focus that has 9 square centimeters and it is propagating. An interesting line of investigation has been this. Actually, this is a project that has been developed for the past 10 years by Dr. Jeff Loeb here in Michigan. There are cerebral regions that have frequent interictal discharges. They show they have the activation of a specific biochemical pathways, and this biochemical pathways is called the MAP kinase, meaning microtubule associating protein kinase. This biochemical pathways is known to increase synaptic connectivity between cells. So there is in the regions of the brain with a lot of interictal discharge, epileptic discharges, there is overexpression of biochemical pathways that is known to increase connectivity between cells. And epileptic discharges are thought to be due to increase interconnectivity between neurons. So here perhaps we have found something very important. The remarkable finding was that this biochemical pathway has been studied by many years by oncologists, and in some way it is involved in the process of metatasization of cancer cells. So there are drugs that are being used in humans that are blockers of these biochemical pathways. And some of these drugs have been used in animal models of epilepsy. And guess what is the effect? These drugs have a wipe out, they eliminate completely in these animals that have epilepsy, eliminate completely any epileptic form activity. So this is an exciting, interesting finding that is likely to have a future in the next few years.
That's great. That's great to hear. So we are getting close to our finishing time, so I wanted to make sure we have a chance for people to ask questions if they have any. Does anyone have any questions about anything we've talked about so far? Since we have a little more time, one question I wanted to ask was about hippocampal sclerosis, which is basically a loss of neurons in the hippocampus. This is something that's common in people with epilepsy, and in some ways it may be a cause of epilepsy, or it may be a consequence of epilepsy, or it may be both. So if you could talk a little bit about that, and then also we know that hippocampal sclerosis can contribute to memory and mood problems because the hippocampus is very involved in both memory and mood. And we also know that there have been studies that show that exercise can promote hippocampal neurogenesis, so which is the growth of new neurons in the hippocampus. And so So basically, ultimately, my question was, can we actually improve mood or memory or seizure control through exercise via the process of hippocampal neurogenesis?
It's a fantastic question, and I am not sure that I can give you an answer.
Okay.
The best person to answer this question, I forgot his name, is one of my senior colleagues, is the co-director of the Epilepsy Center at University of Michigan, and he deals
Dr.
the neurogen. Yeah, exactly.
Yeah, okay.
Yes, he's the one that does this and is able to do that, to tell you about this.
Okay, great. Well, can you talk in the last couple of minutes about some of your specific research that, and then also any other research around the world that's got you, what's got you most optimistic about the future of epilepsy treatment?
I am interested in this question, the transition between between seizures acute seizures in the normal tissue into chronic epilepsy. And so I have set up a recording system to detect epileptic discharges from a brain slice in vitro. So the slices are stimulated with a proconvulsant, like researchers have done for decades, but the novelty of the project I'm running is this. I am doing recording from 60 different extracellular electrodes, while the majority of research that has been done, and which has been done by studying one cell at a time. So if you can record from 60 electrodes at the same time, so then you can study the spatial distribution, where, how epileptiform discharges originate on a brain slice, and how they originate, how they evolve in time. So I briefly mentioned micro discharges before. So I reproduce the data on micro discharges. Now, the novelty of the technique, however, has led to no very interesting findings. So in my experiments, I see multiple independent microscopic discharges from very closely spaced zones. And when one does a prolonged stimulation, there is a gradual and progressive coupling of these microscopic discharges into a larger zone. So this would be like an example in which in the 2 hours of experiment, then you can go from the small microscopic discharges into a large discharge zone. Perhaps on a larger scale, that's what happens when someone has an initial lesion in the brain for a bleeding or a head injury. So there may be around the area of extravasation of blood, there may be small micro discharges. And like in the experiments, in a couple of hours, there is a progressive coupling in a larger discharge zone. And so within 3 to 5 years, if this is multiplied by 3 to 5 years, then you end up establishing an actual epileptic focus that produces symptoms that can be recorded on the scalp. So the fact that you can study that in vitro means that you can define the details of the mechanisms and you can study a lot of drugs. Yes.
Wow, that's impressive. And anything else in terms of other research that's been being done around the world that really grabs your attention?
Yeah, there is something that really grabbed my attention, and it's something that applying that in humans is a problem. But nonetheless, let me tell you the story, and maybe down the road, this can have a future, because in these experiments that I've seen epileptiform activity in an epileptic animal going away completely with the animal, entirely and totally cured, basically, let's say. So genome therapy. So people have used viruses, plasmids to make cells express some specific molecules that have therapeutic properties. So a human subject that doesn't have a gene and the lack of that gene will cause a disease, well, you can use a benign virus, that the virus gets inserted into the genomes, and then it will produce that enzyme, that molecules, that the deficiency which cause the disease. So molecular biology techniques. You can make a virus that has attached the genome of a potassium channel. So the activation of potassium channels has an anticonvulsant effect. The new drug approved by the FDA, Potiga, is a potassium channel opener. So here you make a virus that when it is expressed, it also causes the overexpression of potassium channels. So the virus infects neurons, inserts in the nucleus of the cell, and when doing so, it also replicates, but it also produces potassium channels in that cell. So the cells infected produce potassium channels, and that allow a decrease in the epileptic discharges. So in the animal models, it worked in a spectacular way. And so the animal was going from, within a month, within 3 to 4 weeks, it went from having many, many seizures a day to having no seizures at all. And there was no impact on the neurological function, at least for what you can test on neurological function in a rat. So basically the animal was cured. So of course it is premature, and the implications and ramifications in this in human subjects are big issues, and probably would not be for the average epilepsy case. But there are desperate cases of human subjects that have catastrophic epilepsy. And in any case, even with the current tools that we have available, they might probably be bound to a very short life. And perhaps in 3 to 4, 5, 10 years, at least these individuals, this type of therapy might be available. Anyway, there is a lot of work to do. But this is something that I think is sensitive to follow.
Sure, and so that would be, if you wanted to read up more on that, that would be, is that viral vectors? Is that the term?
Yes.
Okay, great. Well, it is 7:30, so does anyone else have any last questions? Okay, well, I want to thank you, Dr. Serafini, for covering such a wide range of topics and such a difficult topic, and doing it in the way that I could understand, and hopefully everyone else could understand as well. So, okay, thanks again, and thanks for everyone for
Thank you for inviting.